about my experience testing the MinION Mk1D here in Alaska. First, I want to give a brief land acknowledgement and note that our Trothieta campus at the University of Alaska Fairbanks is located on the ancestral lands of the Dana people of the lower Tanana River. So a little bit more about me and my experience with sequencing technology. So as an undergraduate, I was using Sanger sequencing as a tool for understanding phylogenetic relationships. And then as a graduate student, I moved on to next generation sequencing as a tool for understanding questions in population genetics. And then about 9 years ago, I joined the MinION Access program, the MAP program, for nanopore sequencing. And I've been using nanopore sequencing ever since. So just a little bit about my background using these kinds of tools. So one of my passions is in research is genomics and using genomics tools. And one of the things that I love to do is share that passion with students and help train the next generation of researchers. And I found that the MinION sequencing platform is a great way to introduce students. It's a gateway to genomics. And so I've been using things like MinION hackathons as tools or experiences to give students genuine research experiences performing sequencing experiments in the lab using this, putting the power of sequencing right in their hands. And so when Oxford Nanopore asked me to beta test their most recent MinION device, the Mk1D, I was really excited. I jumped at the chance. And so today I'm going to share with you some of my experience testing this both in the laboratory and one field test that we ran. So to give you an outline here, so we ran the Mk1D in a standard laboratory environment, as well as taking advantage of the extended ambient range conditions of this new device. So it has operating temperatures that are wider than the Mk1B. And so here in Alaska, we're often challenged by cold environments and interested in doing sequencing in the field. And so we tested this device in colder environments and the field test. So what does the laboratory environment test look like? So here you can see Danielle Wren, a graduate student in the lab, loading the Mk1D there. We had it hooked up to our lab server. And so we prepared a ligation sequencing library kit, a kit 14, and with the lambda control DNA as our template. We loaded this on an R10 flow cell and ran this using live base calling, using the super accurate base calling model for 18 hours on our lab server. So a pretty standardized experiment that if you ever used this kind of technology, maybe you may have done as one of your first runs as well. So what kind of data did we get out of this? Great. So during these 18 hours, we received about 500,000 reads. That was almost 7 gigabases of high quality data. This is data above a Q-score of 10. If you look at the distribution on the right, you can see that there was a lot of data that had that was a very high quality with a mean or a median in the mid 20 range for this initial testing run. I wanted to make sure that the Mk1D was operating in standard laboratory environments, operating as we expected. So during the sequencing run, we kept track of the translocation speed, the sequencing speed. You can see on the graph on the left that the sequencing speed was sitting there right around 400. This is just as it's in its optimal performance range and what we expected, right? This is a laboratory environment. It should behave normally. But before we tested in any kind of different environments, we want to make sure it was working for us. Additionally, the operating temperature, you can see on the graph on the right, was sitting there right at 35. So even though the lab, we had a temperature probe set up in the lab monitoring the temperature right next to the device, but the device itself was measuring 35 degrees C. Again, rock solid performance there, and just what we were expecting. You may have noticed that when we were loading the library, there was a Mk1D as well sitting on the bench. So for this set of standard laboratory environment testing, we actually, when we created the library, we split it across two different flow cells, and we loaded the Mk1D and the Mk1B simultaneously. So we had these two experiments, these two sequencing runs going on right next to each other. They were a little bit spaced apart so as to not interfere with one another, but we were collecting data at the same time. And you can see, just as a measure of performance, the quality score of the reads. These are unfiltered reads here being displayed on the right. And you can see these violin plots showing a distribution with a median Q-score around 22 for both the Mk1D and the Mk1B. So this new device, it looks to be performing as well as the device that you may have been using in your lab for a number of years, but it has some improvements, as we'll see in just a moment. So one of these improvements is this increased temperature range. So when I received the device and learned a little bit more about it, I was instantly noticed that the temperature range, the ambient temperature range, dipped as low as 10 degrees centigrade and then as high as 35 degrees Celsius. And so I was quite surprised by this. And here in Alaska, we're often, as I mentioned, we're often challenged by colder temperatures. And I thought, well, let's test this colder end of the range. I'll leave it to the tropical field biologist to experiment at that higher end and look at the colder range. So I thought, okay, we can attach the Mk1D to a laptop, and it conveniently fits into a small laboratory refrigerator, a stand-in for an environmental chamber in my lab. And so I took that refrigerator and adjusted the temperature so that it maintained a temperature around 12 degrees for one test. And then I dropped the temperature below this ambient temperature range so that it was around 3.4 degrees. So at that lower temperature, the temperature fluctuated quite a bit as it got to as high as 8 degrees Celsius, but it did actually dip below the freezing point. So it did dip to -2 degrees or 1.9 degrees Celsius during some of that time. So these were the two environmental conditions where I took the sequencing runs that I had set up for the standard laboratory conditions, and now I was running them at these lower temperatures. And so these were actually the same libraries, the same flow cells that were at the standard conditions. We just popped them in the fridge and ran them at this cold temperature, and then for 18 hours, and then ran them at a colder temperature for another 18 hours to really challenge this device to see how it was performing. So across these three temperature ranges, you can see a measure of the quality of the output data. So the graphs on the right show the average quality, base core quality score for the output for the 23-degree standard test, for the 12-degree test, and for the 3.4-degree test. And you can see that the quality score is about the same. There was actually a slight increase to 24, 23 for the colder temperatures, but this is based on just one test. And I think my response to this was that we're seeing high quality data being generated at temperature ranges that are below the previous model and well below or even outside of the current range of this device. So at 3.4 degrees, we're still seeing good quality data being generated by this Mk1D device. you may have noticed in the picture that we actually had the Mk1D on these colder temperatures as well. So in these tests, we did see an okay performance of the Mk1D at the cold temperatures and the very cold temperatures. Maybe there's a slight decrease at the very cold temperatures. I will note that during the testing, the Mk1D was really throwing a lot of errors. So the MinION software was complaining that the device was not at its optimal temperature range and continuously alerting me to this fact. And when I went and looked at the sequencing speed of these sequencing runs, this is where I noticed that there was a real drop in performance. So for the Mk1D at these colder temperature, that's this brown line at the very bottom of this graph, you can see that it dropped well outside of the optimal range and was in the 250 nucleotides per second range. And this is likely to impact the overall sequencing yield because the templates are just going through the pores much slower. So we did see a difference. Whereas the Mk1D, even at this very cold range, you can see that purple line is right up there within the normal operating speeds, right around 400. So I was very impressed by its performance at these cold temperatures. Next, I wanted to take the Mk1D out into the field and see how it performed in a field testing. So I used essentially the same conditions. I did swap out the template DNA. So instead of using that lambda control DNA, I wanted to use DNA from an ongoing experiment. And so we're surveying Alaskan soils here. And so I used DNA extracted from soil using the QIAGEN Power Soil Pro kit. The DNA was, after extraction, it was spree cleaned prior to library preparation. But then we ran the library prep in ligation sequencing kit and ran it on a R10 flow cell. And then we took this setup out into the field here in Alaska. Here you can see the setup in the field. So we have a laptop connected to a portable battery. So we had our Goal Zero battery hooked up to this just to make sure that the laptop had sufficient power for an 18-hour sequencing run. And so I set the device up out in the field and monitored the temperature. We had overnight temperatures that did drop to 10 degrees Celsius at the time, and there was actually some light rain. So we had an umbrella that did protect the laptop setup because I was not confident of the ruggedness of the laptop. So we had temperatures that fluctuated. It started, more in the 15 degrees Celsius when it was initially set up, and then dropped down to the lower end. The flow cell performed great. We had 512 active pores at the start, which was quite amazing. And so the sequencing ran for 18 hours. Here you can see a graph of the sequencing performance over time. And even though the temperature was dropping constantly during this test, we saw no change in the sequencing output during those initial 18 hours. So you can see the yield just growing pretty consistently, both in terms of gigabases and reads. This is pretty optimal performance, what I would expect in a laboratory setting, but here we were sequencing in the field. What kind of data did we get? So this was soil DNA that we did use bead beating to crack open the cells and to extract the DNA. So we didn't have super long reads here, but that was to be expected. The median read quality of this data was 22.7, so really high quality data. Again, this is unfiltered, so this is looking at all of the reads generated. In that 18 hours, we generated over 10 gigabases of data, of which 7.8 gigabases were above Q20. So I was really happy with the kind of output that we were getting from this sequencing run. I mentioned that this was data that was generated from DNA from our Alaskan soil project. And so what the kind of data that we generated here with read lengths that are above 4 kb, this is allowing us to develop seriously interesting biological insights into the soil microbial communities that we're surveying. So we're trying to understand, with data like this, we're trying to understand soil health through the lens of genomics. And so to establish a baseline for high latitude soils, we have a collaboration with the Anthropocene Working Group as part of the U.S. Fish and Wildlife to understand these microbial communities, surveying places like these points are indicating in northern Alaska. And so being able to generate high quality metagenomic data to help us understand things like antibiotic resistance is really important. One of our challenges with this project is getting soil back into the lab. And so having a device that enables more variable field conditions and is field ready at the get-go is a really exciting tool that I'm looking forward to using more in the future as we explore these this kind of project even more. So to summarize my first impressions of the Mk1D, the improvements in the thermal tolerance provide more opportunities for field and resource limited sequencing. So you don't just have to be sequencing in the field, but even in conditions, laboratory conditions, we aren't always able to maintain those 23 degree Celsius temperatures, whether it's getting hot or cold. I've been in both laboratory conditions, and having a device that actively maintains its temperature represents a significant upgrade compared to the capabilities of the Mk1B. I want to thank you for your attention, and thanks for ONT and the team there for giving me the opportunity to beta test this. And thanks Danielle, Aggie, and Bevan for helping the lab preparing all of the samples for sequencing and letting me share this information. And next, I'll turn it over to James.
Oh, thanks, Devin. Hi, my name is James Platt. I am the Director of Sequencing Product Management here at Oxford Nanopore, and today I'm going to give you an introduction to the MinION Mk1D. So the Mk1D, it really is the next step in our goal at Oxford Nanopore to enable the analysis of anything by anyone, anywhere. The Mk1D enables, like Devin showed, anyone to sequence themselves in the lab or to take it out to the field where the sample is and really take control of their study timelines. So before I get into the detail, I want to just give some background. So the MinION was the first device that we launched at Oxford Nanopore, and that was 10 years ago now, which is incredible. But the platform has changed a huge amount in those 10 years. It's got so much better. It's come on leaps and bounds. And we've learned a lot in that time. But the MinION itself has largely stayed the same. In the time since launching the MinION, we've launched the GridION, the PromethION, the Minute, the Mk1C, the P2 Solo, the P2 Integrated, and more recently the Elysian. So what we've done here is actually strip the MinION right back and rebuild it from the ground up, and really incorporate all those things we've learned over the last 10 years. And so some of those things you can see here that I'm going to go over, but there's a lot of under the hood changes as well to improve reliability and really improve customer experience. So one thing we've done is make it really easy to install. We've really thought hard about the installation journey, and it's even easier than the Mk1B to install. It now comes with USB-C connectivity, so you can use it with modern laptops. It's got really nice visual status indicators on the back. You can see here annotated, and what these do is give you feedback about what's going on the device at the time, so you don't have to go into the computer and open Minnow to see what's going on with the device. The big thing that we've improved here, and Devin spoke quite a bit about, is this active temperature control. So we've really opened up the range of environmental conditions that you can use the device in, and I'll go through that in a bit more detail in a couple of slides' time. And then towards the end of the talk, I'll talk about some of the applications that the MinION enables for sequencing of small genomes, metagenomics, and targeted approaches. So head-to-head here, Mk1B versus Mk1D. So we've got pictures on the side here that are showing the two different devices, and you can see phenomenally our engineering team have been able to make the MinION even thinner than it was, which is something, and put in active temperature regulation in there too. So like the Mk1B, the Mk1D runs MinION or Flongle flow cells. The Flongle flow cells is with the addition of the Flongle adapter. As I mentioned in the previous slide, we've really improved the temperature control here. The connectivity, we've moved from USB-A to USB-C, which really enables people running modern laptops to run the device. So, for example, my computer doesn't have any USB-A ports. We've also got a nice IT requirements document, which really helps you choose a suitable computer to run the device. And also, we've designed the Mk1D to be compatible with the iPad Pro. So I'll talk about a little bit at the end. We've got in the works an accessory that enables you to run the Mk1D off an iPad Pro. And yeah, then we've got the status LEDs, which I mentioned in the previous slide. So really giving you that at a glance look at what's going on the device. And then we do on the community have a wealth of information about how to get started with MinION and how to get up and running, and all protocols and how know-how documents. But we also do offer training at an extra cost. So if you're really keen on getting started as quickly as possible, you can sign up to that training at an extra cost. So moving on to that active temperature control that I spoke about. So the Mk1D really does have significantly improved thermal regulation, and it really puts the anywhere into anything, anywhere, anyone. So because we've got this onboard Peltier, it means we can extend the range of ambient temperatures we can support. So we've extended it to 10 to 35 degrees. And so what we've got on this slide is a couple of bits of data, and I'll walk you through these bits of data. So what we did is an experiment where, similar to what Devin showed, but we did it over a much wider range of temperatures. So we've got an incubator essentially where we can control the temperature, and what we did was place Mk1Bs and Mk1D devices in the chamber, and we changed the temperature ranging from 5 to 50 degrees C, and we performed sequencing at each at 5-degree intervals. And so what we've got on this slide is a plot of what happens to sequencing speed. And so in this experiment, we were running R10 chemistry, and for that we have a target sequencing speed of 400 bases per second. And on this plot, you can see two sort of horizontal lines, and this is 10% above 400 bases and 10% below 400 bases. So this is the ideal speed that we want to be sequencing at. And what you can see here, when we did the experiment for the Mk1D, we were stable across the broad range of temperatures here. It's very stable across the supported 10 to 35 degrees C. Whereas the Mk1B, as soon as we got out of this sort of standard laboratory conditions that we support, the 18 to 25 degrees C, you can see that as you get colder, the sequencing speed got a lot slower. And as you went to higher temperatures, so for example, maybe you're in a hot lab in the summer, or in parts of the world where air conditioning isn't commonplace, or outside, we start to get higher sequencing speeds. And what does this really mean? So why do we care about this? And there's two reasons that we really care about that sequencing speed and tightly controlling it. And that's one is because we train our base calling models on the data that is at 400 bases per second. So that gives us the highest, if we're sequencing at the correct speed, it gives us the highest quality base calls, highest quality data. but also we sequence at the right speed, we get the most data out of the flow cell as well. If we're sequencing at too slow a speed, we just won't get the DNA or RNA molecule just won't go through the pore as fast, so we just don't get as much data. So you can see on the bottom here that across the range of supported temperatures on the Mk1D, we're able to get really high quality data. So this plot is showing you on the y-axis is modal Q-scores, and then on the x-axis is the temperature range. So whereas the Mk1B in our experiment, as soon as you got out of that standard laboratory temperatures, your quality started to fall off. The other benefit that we have on the Mk1D is that it's because we've got this active temperature control, and we, like Devin was saying, we have this a temperature that we sequence at, and to get to that temperature, it typically takes a bit of time to warm up the flow cell to get to that temperature. And so what that means is that when we're setting up an experiment, when either if it's a flow cell check or a sequencing experiment, because we've got active temperature control, we can ramp up that temperature to get to the set point quicker on the Mk1D compared to the Mk1B. So what does that really mean in reality? So here we've got a table comparing the time it took to get to set point. So on a Mk1B, it takes 5 minutes. Mk1D, it just takes 1/10 of that time, at half a minute. The gridiron for comparison takes 3 minutes to get to the set point. So we've made an advance there over the gridiron. And how that translates to the total time to do a flow cell check, it took 12 minutes on a Mk1B and 7.5 minutes on a Mk1D. So we saved 1/3 of time there on a flow cell check. The other thing to mention is during a sequencing run, because you're getting to the temperature quicker, it means you start getting data off your flow cell quicker. So for those kind of applications where time to answer is important, getting that answer a few minutes sooner is going to be a good thing. Okay, so then shifting away now, what I'm going to do is talk about what you can do with the technology. So as I mentioned before, you can run MinION flow cells or you can run Flongle flow cells on the Mk1D. I'm going to focus on MinION flow cells and what a MinION flow cell can do. So the flow cell itself is incredibly flexible and versatile. It can be used for a variety of different applications and read lengths. So with nanopore sequencing, you're really doing real-time single molecule sequencing of native DNA or RNA molecules. So that means you get direct methylation or other modified bases in the data for free. You can sequence RNA directly without having to make a copy. And the platform itself is read length agnostic. So that means that as long as you can get the nucleic acid molecule, you can get adapters onto it and you can get it onto the flow cell, you can sequence it. So you can get read lengths from 20 bases to megabases in length. It's really very flexible in that respect. So then going on to the kind of things you can do with it. So obviously you can sequence native genomic DNA on the flow cell. Depending on the application, you could look to get somewhere between 15 and 35 gig off a flow cell. We've got different multiplexing options. So we've got up to 96 barcodes with either a PCR or a PCR-free barcoding approach. So you can barcode samples and still maintain those modifications on the DNA. So one of the things you can do with multiplexing is sequencing of full-length plasmids. So you can get highly accurate plasmids off a device from as little as $13 per plasmid. Another nice feature of the technology and the flow cell is the real-time nature of the data generation. So as soon as you've generated enough data from your experiment, you can stop it. And you can then reuse the flow cell. So a flow cell is a bit like a battery. It has a sort of capacity for sequencing. So if you only need a few gigabases worth of data off a flow cell, you can and there's still life left in the flow cell, you can stop the run, wash the library out, store the flow cell, and come back to it another day and reuse it with a completely different library. Cool. So then as I mentioned, the platform is really versatile, and one of the areas the MinION platform excels because of the output that it gives is microbiology and infectious disease. So I'm just going to run through some of these applications that you can run on the device. So one of the first one, bacterial isolate sequencing with MinION, enables you to rapidly characterize outbreaks or look at antimicrobial resistance and do things like molecular epidemiology. You can also do full-length 16S sequencing, which provides you with a high level of taxonomic resolution than you can typically achieve with sort of partial 16S analysis or legacy Sanger sequencing. The MinION Mk1D here really represents a really cost-effective and small footprint device to do these kinds of analyses. Moving on, viral amplicon sequencing. So in recent years, the MinION and GridION platforms have been really important tools for viral amplicon sequencing. It's been widely used to track variants and identify novel variants, and then inform vaccine development and guide public health decisions. So you may remember that the MinION was really instrumental in the SARS-CoV-2 response. A lot of sequencing happening on MinION flow cells for that. It's also been used in a variety of other outbreaks as well, including Ebola, Zika, and Mpox. And then finally, metagenomics workflows really enable labs to perform analysis of complex mixed samples and detect a wide variety of pathogens and profile antimicrobial resistance, genes or plasmids without really prior knowledge of what's in the sample. So it's really excellent. And then last slide with some data on, we're just showing you here an example of what you can do with the data and the quality of the data that you get off a MinION flow cell. So on the left, we've got some internal data that compares some assemblies of a metagenomic sample. And what we're doing is on the top, we've got well, here we've got two plots. They're both bandage plots looking at the quality of the assemblies. And so we're comparing on the top short reads and on the bottom nanopore reads. And you can, so what you're looking for in one of these bandage plots is a single assembly, with a sort of complete unfragmented assembly. And so what you can see with the short reads at the top, you get this sort of hairball, highly fragmented, incomplete genome assemblies. Whereas at the bottom, you can see nanopore reads are giving you single, complete metagenomic assemblies. And the benefit as well is you can get these really rapidly with nanopore sequencing because of that real-time sequencing. And then on the right, we've got some data from Dr. Ryan Wick, a bioinformatician at the Doherty Institute in Australia. So here, this is from a blog post where he's benchmarked the base caller that we use for converting the raw signal into base calls. And he's basically looking at our 4.3 base calling models. So these are the ones that are in Minno right now, so available to everyone. These are not the research models. These are the ones that are widely available in the software. And what he's showing is he's looking at a metagenomic population, and he's looking at the genome assemblies that came out of that metagenomic population, and he's looking at the consensus accuracy. And what you can see across the different species that are present in the sample, that we're getting really highly accurate assemblies here, and a lot of them are actually over Q60 in terms of consensus accuracy, which is really, really phenomenal. And just to reiterate, this is just with nanopore only as well. And then, so final slide, I want to say that over the summer, we've had the Mk1D out with beta testers and out with closed early access customers. So Devon was one of those beta customers, and we thank those customers for really putting the Mk1D to a test in the field. and stress testing it and giving us really good feedback. So now we're really excited to be at a point where we can start opening that up to more people and shipping more widely, which is really exciting. So in November, we're looking to move the Mk1D into an open early access, and that will be happening from the Wednesday, the 13th of November. And at that point, we're going to begin shipping devices to customers. And so what we're doing is we're going to provide an exclusive upgrade offer to existing MinION customers, and that this will take the form of an email that will be sent out. So anybody that is an existing MinION customer will automatically get emailed this. And also, if you register your interest through the link here on the slide, or scan the QR code and fill your details in, you'll also receive that exclusive offer to be first in the queue to get a MinION Mk1D. And then the other thing I just wanted to mention here is that iPad accessory that I mentioned earlier. So this is coming soon. We're working really hard on this. So this is an integration between an Apple iPad Pro and the Mk1D, enabling us to take advantage of that Apple ecosystem. We're expecting to be available in 2025. And if you do want to hear more about that, again, you can go and scan the QR code and fill your details in. Okay, so I hope you enjoyed the presentation, and where I've told you about the new MinION Mk1D and some of the powerful genomic insights that you can achieve with such a small but really mighty device.